Okay, good evening. I'm Dr. Robert Ife, and welcome to the Chem and Biochem 2020 Fall Webinar Series. We have an awesome talk, first talk in November, and my job today is to give us the ground rules for this evening's webinar. You'll notice that there's a couple of students and faculty in the Zoom chat today. Our speaker has graciously suggested for you to interrupt her at any point that you want with questions. So this is a new one. Don't wait till the end. Please ask questions throughout. And if you're on YouTube watching, you are free to email at fall.webinar.questions@gmail.com, and I will ask on your behalf. So let me introduce Alyssa Dubord, who is going to say a couple of quick remarks about this evening's speaker. Alyssa.
Hi, my name is Alyssa Dubord. Today, I would like to introduce Morgan Hawker. She obtained her BS in chemistry from UC Santa Cruz and then went on to obtain her PhD in chemistry, specifically in materials chemistry and chemistry education at Colorado State University, and then went on to obtain her postdoc at Tufts University in training and education and critical research skills in the lab of David Kaplan. And presently, she is an assistant professor at California State University, Fresno. So please join me in welcoming Morgan Hawker.
Thank you, everyone. And as was alluded to, I want to start out by saying that this is a very interactive presentation. So I encourage you to participate to the absolute fullest extent that you're able to in this virtual format. And I welcome any questions or discussion during the talk. So please feel free to give your input or ask a question at any time. So I joined the chemistry department at Fresno State about a year ago, and we're very much still in the process of creating a functional research space. So as such, the story I want to tell you today is really an overview of the motivation behind our work. So kind of like a behind the scenes look. So toward the end of the story, I'll show you some very exciting hot off the presses data and kind of share where we're going next. So I told you the seminar would be interactive, so I'm going to start off with a question. Feel free to type in the chat or use your reaction features if you're in Zoom. How many people in the audience wear contact lenses? Okay, few. If you do, have you ever thought about what your contact lenses are made of? Not quite as many. So many contact lenses are made up of a soft silicone-based polymer, and the chemical structure for that shown here. And the reason why this polymer is used is because it's optically clear, it's flexible, meaning it can conform to the surface of the eye, and it also allows for gas permeation. However, the properties of this polymer are unfavorable when it comes to interacting with the watery surface of the eye. And this can ultimately lead to eye irritation, which nobody really needs in their life. So the reason why these silicone materials are so widely used as contact lenses is because their surface can actually be modified. And this surface modification can lead to a more favorable interaction with the eye's surface. And as such, preventing irritation. So the story that I want to tell today is related to this idea of modifying the surface properties of polymer materials for use in biomedical devices. Now, by biomedical devices, I mean anything that's implanted in or contacting a biological environment, whether it be a Band-Aid on the surface of the skin or something like a hip joint replacement. I'll first introduce polymers and plasma, including what they are and their potential use to fabricate biomedical devices. Then I'll discuss one specific polymer that my group is currently researching for biomedical device use, and I'll tell you a bit about our motivation behind why we want to use this polymer. And lastly, I'll finish up by showing you some interesting findings from our preliminary studies with this polymer thus far. you'll notice that this story is actually composed of many questions, and I hope to convince you all of the importance of plasma modified polymers as biomedical materials by the end of this talk. So you'll have to give me some feedback, but hopefully that will be a successful end to the story. So biomedical devices are critically important for the betterment of human health. You may be familiar with some of these examples, like joint replacements, stitches, or staples. Some are a bit more exotic, like tissue engineering constructs. Here, healthy cells are grown in a petri dish, generally on a material scaffold. And then this scaffold with the healthy human cells can be added to an area of damaged tissue. The idea is that as the scaffold degrades, the new cells merge with the existing human tissue, resulting in tissue recovery. Another key area of biomedical devices are those for drug delivery, and these can take many different forms, some of which you may be familiar with. Here we're showing things ranging from microporous scaffolds to more of a micelle-like structure, even to polymeric nanomaterials and hydrogels. So regardless of the type of biomedical device, they're generally made, constructed from three different classes of materials, metals, ceramics, or polymers. And in some cases, the application necessitates using more than one material. So if you look up in the upper left corner at this finger joint replacement device, you'll see that it's made of both a metal for wear resistance or anti-corrosion properties, as well as this polymer kind of cap coating on the material. And this is because you're bending this joint over and over again. So you really want it to have low resistance during that entire process so that the motion is very fluid. So polymers are one of the most popular classes of biomedical materials. And you might be wondering why, but before we get there, I want to give you an introduction to what polymers are for those who might be unfamiliar with it. Every polymer starts from one or more monomers. Think of the monomer like a base unit. Polymers are formed when a monomer undergoes a chemical reaction that covalently links the monomers together. And this could be a single monomer or multiple different types of monomers. I'm showing a single monomer here. For example, the monomer ethylene, shown on the left, is used to make the polymer polyethylene. The subscript N implies that this is a base unit repeated multiple times. Now, it's not uncommon for polymer molecular weights to reach hundreds of thousands of grams per mole. So these can be quite large molecules. Multiple polymer chains interact with each other, which yields this bulk polymer material. Chains can either be physically or chemically cross-linked to one another, depending on the material. So going back to polyethylene, the way that the chains are cross-linked impacts the resulting properties. Here are two different polyethylene materials that you may be familiar with. On the left, the high-density polyethylene, and on the right, low-density polyethylene. These materials have drastically different properties in terms of their toughness and flexibility, even though they share the same polymer backbone. And this polyethylene example really illustrates why polymers have so much utility as biomaterials. Going into a bit more detail, this figure here shows the Young's modulus on the y-axis here of biological organisms on the left in orange and common biomaterials on the right in blue. So what the Young's modulus is essentially is a measure of the mechanical properties of a given material. So lower values down here toward the bottom of the chart correspond to softer materials, and harder values in the upper portion of the chart correspond to harder materials. So you can see biological material or biological organisms over here on the left side. So fat and cartilage are softer types of organisms up here, and we have bone and tooth enamel, much harder materials. So over here on the right, essentially all of these materials ranging from nylon down to alginate hydrogels, these are all polymer materials. And this range captures why polymers are so essential for use in biomedical devices. So if the mechanical properties of the tissue or part of the body that you're trying to mimic with the biological material, the biomedical material, if those mechanical properties match, then the material itself is less likely to cause inflammation, and inflammation can lead to device rejection. So if you're trying to repair a tissue, the last thing you want is for that material to be rejected by the body. That does not promote healing. So although polymers have this excellent range of mechanical properties, they do have some challenges, and I'll talk about those next. So polymers typically lack any type of specific interaction with a surrounding environment, whether that be biological or otherwise. And these are when placed in the body, these polymer materials are sometimes referred to as being biologically inert. Now, this can be incredibly problematic because almost all types of biomedical applications require the surface to interact in a very specific and targeted way with its surrounding environment. For example, to create a tissue engineering construct, it's important for mammalian cells to attach to that polymer material to allow for the tissue to regenerate and reform and regain its function. You may also consider applications like wound healing, where you need concurrent cell attachment, but also the prevention of bacterial attachment. So no matter which way you look at this, having specific control over how the environment interacts with the polymer is absolutely critical. So to realize this entire range of biomedical applications, whether it be drug delivery, tissue engineering, or any other type of environment where you want to induce a specific outcome between the biological environment and the material, it's necessary to control this interfacial region between the material and its environment. So controlling the interface can be achieved through modifying the surface of the polymer biomaterial. And this generally means to add specific types of functional groups onto the polymer surface that either promote or prevent a particular type of desired interaction. So there are numerous different techniques that can be used to modify polymer surfaces. But my research is specifically focused on a strategy called plasma modification. So plasma, sometimes called the forgotten state of matter. Okay, so this is the fourth state of matter, and it's a partially ionized gas. It's actually a mixture of neutral gas molecules or atoms, ions, radicals, and free electrons. So think about it kind of like a vegetable soup of gaseous chemical species. So you might be wondering what's so great about plasma treatment. Well, first of all, the type of plasma modification that we do is effectively carried out at room temperature. What this means is that it can be used to modify many different types of substrates, including polymers that are somewhat delicate, without damaging their advantageous bulk properties that I talked about earlier. Also, because plasma treatment is a gas phase modification technique, it eliminates the use of any solvents. And this is especially key for polymer modification because many different types of polymers are incompatible with different solvent processing conditions used in alternative modification techniques like wet chemical modification. So this is showing a picture of a plasma reactor similar to the ones that we have in my lab. And you can see this characteristic blue glow, which is arising from excited state gaseous species emitting visible light. And you can think about this kind of like a giant light bulb, okay, except the electrode is actually on the outside. So this is an inductively coupled plasma reactor. So let me tell you about a little bit more about how it works. Okay, so here's a schematic of the inductively coupled plasma reactor. The first thing that we do, and this is a tubular cylinder. So the first thing that we do is open this plasma reactor. There's an O-ring joint here, and we can add in the polymer material we want to modify anywhere in the chamber. So next we use a mechanical pump to create a low pressure environment inside of the reactor. After that, we flow in a gaseous precursor. So this could be like a pressurized gas cylinder, or it could be the vapor from a high vapor pressure liquid. And this gas that we flow in is referred to as the plasma precursor. And really the precursor can be essentially anything that we can get into the gas phase. This could be something simple, relatively simple, like molecular oxygen, water vapor, short chain hydrocarbons, even complex, like more chemically complex things like essential oils. Okay, we can get huge variety in this precursor. Once the precursor has been added, we use an RF power supply to apply power to this induction coil. And what that does is it inductively couples to the gas inside to generate the plasma, that partially ionized gas that's a mixture of neutral gas molecules, ions, electrons, radicals, etc. So as I alluded to, plasma treatment is highly customizable. For example, we can change where we place the substrate relative to the induction coil. You saw in the previous slide that the plasma glow is very intense in this region closest to the coil and tends to be more diffuse further away. So that can have important implications on the modification outcome. We can also change the precursor itself, what that gas is that we're introducing. We can adjust the pressure by changing the amount of gas added to the reactor. And we can change the applied power, so applying a higher or lower radio frequency power, higher or lower amplitude, I should specify that, or higher or lower applied power. And then lastly, the time. So how long the material is exposed to the plasma. That has important implications as well in terms of the resulting modification. So by carefully selecting and modulating these key parameters, it's possible to achieve different types of outcomes regarding what happens to the polymer surface upon plasma treatment. It's possible to activate the surface, which means essentially adding specific types of functional groups onto the surface. It's also possible to deposit a thin film on the existing polymer surface, and this is referred to as plasma-enhanced chemical vapor deposition. Lastly, it's possible to etch the polymer surface, which removes some material. Now, plasma chemistry is incredibly complex because of the range of the gas phase species that can be generated, but also their energies. So both the relative quantities of these different types of species and their energies will ultimately dictate which of these results, what the end result is of the surface modification. And as you could imagine, there's sometimes competition between these different types of processes. So for example, a particular set of plasma conditions could result in deposition and then etching of that deposited film, depending on the relative concentration of different species in the gas phase and their energies. So as an example, I'll show you a process that's heavily simplified to illustrate how the deposition of a film can occur in the plasma. So I shared previously that plasma, one of the species found in plasma, the partially ionized gas, are positive ions. So what can happen is that these positive ions can bombard the surface and leaving behind this so-called active site. So this could be a missing atom, this can be a missing electron. We're just talking about a generic kind of active site that's present on the polymer surface. So next, the plasma can also generate radicals from the precursor. And this again depends heavily on what the precursor is and the different plasma treatment conditions. But let's say that we have a precursor that generates substantial quantity of radicals. And what can happen is these radicals can interact with the active site, thereby building this film on the surface. And I'm showing these processes happening in a stepwise manner, but in reality, they're all occurring concurrently throughout the plasma treatment process. So this is just to give you a kind of illustration of a possible type of process. But again, there's multiple processes occurring within the plasma. And when that happens, it influences what happens to the surface itself. So at this point in the story, I hope I've convinced you of the importance of biomedical devices and also the broad utility of polymers to create those devices. So now I want to show you some proof of concept work that I did before I came to Fresno State to illustrate the potential of plasma modifying polymers to change their interactions with a biological environment. I definitely want to pause to make sure that I will be able to answer any questions that anybody has. So please feel free to unmute yourself or type in the chat at any point. I'll be more than happy to answer your questions. All right.
What kind of pressures are you working at typically?
Yeah, so we tend to work in the 50 to 200 millitorr range, depending on the precursor, and that's above the base pressure of the mechanical pump. So, yeah. Any other questions?
I have a question.
Yeah.
Formed, maybe. So you talked a little bit about the wanting to create modified surfaces for specific purposes, or, to be a good interface between either the material and the biological environment. But then you're also talking about all of these variances and complications that come with the modifying. And I'm just wondering what how much variance is there? Obviously, there's some that you have to be concerned about it, but not so much that it's actually interfering with you carrying out what you're aiming to do. That's a good way.
No, that's a great question. And there's a lot that comes along with developing a particular plasma modification strategy. So I should, back up and say that once we apply a strategy, so let's say we're using a water vapor precursor at a certain pressure and a certain applied power for a certain time. So once we control all of those variables, the outcome of that particular modification is reproducible.
Okay.
So we're able to reproduce a particular effect over and over again on the same type of material. But what I was illustrating there with the different parameters that we can change is that we have a lot of knobs to turn and buttons to push, so to speak, and literally, in terms of controlling what those gas phase species are and what their energies are, that can then in turn change what the surface looks like. So a lot of what we do is, for example, taking that water plasma precursor, what would happen if we used a higher power versus a lower power? Well, we do see differences in those surfaces.
Okay.
That
was a good answer. Thank you.
Awesome. Other questions so far? All right. Well, don't hesitate to interrupt me. All right, so before I came to Fresno State, I was working on my PhD at Colorado State University, and there I worked with a polymer called polycaprolactone, or PCl. And this polymer is very popular because it hydrolyzes into non-toxic byproducts in vivo, meaning it does degrade as a function of time. So for this research, I fabricated three-dimensional polymer scaffolds. And the idea behind this fabrication was to create a device that mimicked the extracellular matrix. So on the bottom left here, I'm showing a scanning electron microscopy image that demonstrates the interconnected porous structure of this PCL scaffold, which is at least similar to the extracellular matrix in terms of its three-dimensional interconnectedness. So think of this like a very tiny version of a kitchen sponge, okay? So the problem with this polymer is that it's incredibly hydrophobic. And this is an image taken using a technique called water contact angle goniometry. So a water drop is placed on the surface and the angle between the drop and the surface is measured. So what you're looking at here is a side-on view of the water drop on the surface, showing it beading up, similar to what you would see in like a Teflon pan or on the surface of a Gore-Tex raincoat. So the goal of this project was to enhance the hydrophilicity of the PCL scaffold to enhance the cell attachment on the scaffold surface. So I took an approach where I actually used two different plasma precursors, one allyl alcohol and the other allyl amine. And I studied the effect of treating the scaffold with each precursor independently, as well as the precursors in different mixtures in the gas phase. So creating this mixed gas system, and that's referred to as plasma copolymerization. So I was interested in tuning the surface wettability and measuring how that impacted the interaction between the surface and mammalian cells. But another really interesting kind of unintended piece of this study was to also vary the quantity of nitrogen containing functional groups on the surface. So I'll talk about that and how it connects to, or is related to the differences in wettability surface. So after treating the scaffolds with the different plasma precursors, I used contact angle goniometry to measure how water interacted with the scaffold. So remember that the water drop sat on top of the scaffold before it was exposed to the plasma. After plasma treatment, I'm showing in the video an allyl alcohol modified scaffold. I observed that the water drop was absorbed by the scaffold, and this is shown by the looped video. So this demonstrates that we've effectively enhanced the surface hydrophilicity of this scaffold through plasma treatment. And importantly, we didn't see any changes in the three-dimensional network of the scaffold. So we retained that, but selectively changed its surface properties. And it turns out that the water absorption rate decreases as more and more allylamine is used in the precursor mixture. So we still see water absorbing for the 100% allylamine treated scaffold, but it's just absorbing much slower. I also did some chemical, we're gonna watch the video again. I did some chemical analysis of the surface using a technique called X-ray photoelectron spectroscopy. And what this technique is used for is to measure the composition of the outermost surface, the top 5 to 10 nanometers or so, in terms of the elements present on the surface. So what we're looking at here on the left hand y-axis is the nitrogen to carbon ratio, and on the right, oxygen to carbon. Now the x-axis shows percent of allylamine in the gas feed. So over here we have 100% allylamine, and over here we have 0% allylamine and 100% allyl alcohol, and then some intermediate compositions as well. What we're looking at here is that the oxygen to carbon ratio decreases as a function of allylamine in the gas feed, whereas the nitrogen to carbon ratio increases, generally speaking. So these data demonstrate that the amount of nitrogen containing functional groups on the surface increases with the amount of allylamine in the gas feed. And you'll notice that both nitrogen and oxygen are referenced to carbon. You might be wondering why that is. That's because the carbon content was found also via XPS to be effectively constant between the different surfaces. So these data indicate that plasma copolymerization can be used to concurrently tune the nitrogen content on the scaffold surface as well as its wettability. So what I wanted to do next was to look into how adjusting these properties influence the scaffold's interaction with biological species. Remember, that's kind of the goal here, is to change how the surface interacts with biological species. Here I used human dermal fibroblasts, and I evaluated their viability using an MTT assay. Happy to talk about that more offline. But effectively what I did was take plasma modified and unmodified scaffolds and incubate them with human dermal fibroblasts. And after 72 hours of incubation, I measured the viability of the cells using this MTT assay. So what I'm showing here are predicted results because we anticipated that both nitrogen content and wettability could be important factors in influencing the viability of cells. So these are predicted results if nitrogen content played a more prominent role. I would have expected that the allylamine modified scaffolds would induce higher cell viability than the allyl alcohol. However, if wettability played a more prominent role, I would have expected the opposite, so to speak. So the 100% allyl alcohol modified scaffolds would have shown higher cell viability than the allylamine treated scaffolds. Now, what we actually saw, of course, was neither of these predicted results as usual. We actually saw that all treatments enhanced cell viability when compared to the untreated control, which suggested that both nitrogen content and wettability can be used as tools to promote cell viability. At this point in the story, I want to talk about the polymer system that my group is currently working with at Fresno State. So this is different than the PCL-based polymer that I talked about just now. And the polymer that we're using is silk. So now is a little bit more of a chance for audience participation. So what do you think of when you think of silk?
Spiders.
Spiders.
Silkworms.
Worms.
Clothes. Clothing.
Clothes.
It's very strong for how thin it is.
Strong. Okay. Wow, we have a scientifically minded group here. Good. What else? Did we check all the boxes?
Hierarchical structures?
We're getting into even the more scientific hierarchical structure. Excellent. So silk is a biologically derived polymer material, meaning it comes from a natural source. So this could be spiders, silkworms. The silk that we use in our lab comes from a particular type of silkworm. I'll touch on that in a second. But now I'm going to touch on the hierarchical structure. So silk fibroin is the specific protein or biopolymer that we use in our lab. And this has a block copolymer-like primary sequence, which means it has a repetitive hydrophobic sequences interspaced with hydrophilic amorphous domains and N-terminals. So this is looking at the primary structure. So this primary sequence causes the silk to form a secondary structure that's rich in these anti-parallel beta sheets separated by alpha helices and random coils. And these individual silk structures assemble into a fibroin filament, which is coated with a different protein called sericin. And this is the form of silk originating from natural sources such as silk cocoons. So we use the Bombyx mori silk cocoon as our source of material. And I like to think of silk kind of as the Olympic gymnast of the materials world. Okay, so its unique secondary structure results in a material that, as was mentioned, has both strength and flexibility. So you might be wondering how to make a material that can be put in the body from something that starts out as a cocoon. If we just place the silk cocoon in the body, I'm not sure what all we're going to get from that. Okay, it has kind of a fixed rigid geometry. It also contains the protein sericin, which has been shown to have negative effects when interacting with the body. So we actually need to remove the sericin. And to do that, we can boil the silk cocoons in a sodium carbonate solution. And this effectively dissolves the sericin. And upon drying, we can get just the silk fibroin. And you can think about this as looking like kind of a cotton ball type of material. If you were to pull apart a cotton ball, that's effectively what the fibroin looks like. It's this white, stringy, fibrous material. The fibroin can be dissolved in lithium bromide to yield this kind of concentrated, viscous solution, which then can then be dialyzed against distilled water to remove lithium bromide. And the reason why we want to go through this whole process is to obtain this aqueous fibroin solution. And you might be wondering, well, what's the point of having a polymer solution in the aqueous phase? And actually, it opens up a ton of possibilities. So what we could do is transform it into various types of architectures, ranging from two-dimensional films that we could simply cast on glass slides. We can also do some different chemistries to result in, or to create these silk microparticles, or even create three-dimensional sponges, like I had talked about with the PCL scaffolds. So, and all of those will depend on the application, of course. So the last part of the story I'm going to tell today is about two ways that my research group has customized the surface properties of silk using plasma treatment to really solve two unique challenges, or to attempt to solve, I should say, two unique challenges in the world of biomaterials. So we're very interested in using silk for cell contacting applications. Now, this might be for tissue engineering constructs or some other type of material, but we want it to have we want the silk material for most types of biomedical applications to have favorable types of interactions with mammalian cells, generally speaking. So silk lacks specific functional groups, like many types of polymers, on its surface to signal for cells to attach. And surface modification could be one route that we could use to create these signals. So our initial plan was to enhance the hydrophilicity of the silk materials using a water vapor precursor. So here I'm showing an unmodified silk construct with that interface and the biological species, or the biological environment. And our thought was, by enhancing the surface hydrophilicity, we could encourage cell attachment and proliferation on the surface of the material. And my student created a method to use a water vapor plasma precursor. And this method has actually been used in literature to apply to synthetic polymers to functionalize their surface with OH groups, but it hasn't really been used before to modify naturally derived materials, including silk. So another one of our goals with this project was not only to make the surface hydrophilic, but observe how permanent that hydrophilic modification was. So it's very common for, if we're treating a hydrophobic polymer surface to induce hydrophilicity, it's very common for the polymer to undergo a process called hydrophobic recovery. And what that means is that the polymer chains actually reorient after modification, which subsumes the hydrophilic functional groups. And this is somewhat thermodynamically complicated, but the end result is that the surface rearranges either back to its original state or more closely resembling its original state. So we wanted to study whether or not this process occurs in silk, because this could have implications on the shelf life of silk modified constructs. For example, if you've ever had stitches, the doctor generally removes stitches from packaging that keeps the stitches from getting contaminated and keeps them sterile. So if the surface properties of the stitches changed while they were sitting on the shelf in that packaging, that could lead to some undesirable interactions between the stitches and the body, whether that could cause inflammation or something else. We just want to take some insurance and to know that the properties of the material are going to be consistent throughout the time in which we want to apply them. So here are some data from my students' preliminary study. So we observed here on the x-axis, we're looking at the contact angle of the material, and on the y-axis, we're looking at the contact angle. On the x-axis, we're looking at the aging time. So we have here freshly water plasma treated surface, and you'll see the untreated silk has a contact angle around 65 degrees. When it's plasma treated, we observed this dramatic decrease in hydrophobicity. The surface is becoming more hydrophilic. And after this initial treatment, we aged the samples just under ambient conditions. So they're just sitting room temperature, nothing too fancy. And what we see is that the film contact angles appear to increase. We have fewer data points at these longer times, which we're working on establishing or collecting additional technical replicates here. But we saw some evidence of hydrophobic recovery as a function of aging, and the surface appears to plateau around 35 degrees, though we definitely need to continue to collect more replicates. So we're trying to see if these results are more reproducible, and my student is seeing some really interesting results. So a lot of our silk materials have been sitting around for several months due to the lab shutting down from COVID-19, and that's really inspired us to look closely at the impact that aging our silk films has on their cross-linking and what's called beta sheet formation, which is an irreversible crystallization of the silk film. And we think that this beta sheet formation is likely related or has an impact on the film's hydrophobic recovery rate, as well as how it responds to the plasma. So I'm very excited to look about these experiments that are currently underway to explore this further. So a different challenge in the biomaterials space my group is working on is controlling the degradation of silk-based materials. So silk degrades in vivo via a process called enzymatic hydrolysis, in which enzymes interact with the surface of the silk, cleaving peptide bonds and resulting in degradation. So you could imagine that having silk materials that degrade at different rates is really essential. For example, a joint replacement implant that degrades quickly would be a problem. But having a suture that dissolves relatively quickly, like on the time scale of weeks or maybe months, depending, would be desirable. So the goal of this project is to control the enzymatic degradation rate by controlling the surface properties of the material. And we thought that one way to do this would be by plasma depositing a thin hydrophobic coating on the silk, which might act as a barrier and may prevent enzymes from accessing the surface, thereby preventing the silk from degrading as quickly. So that's kind of an illustration of our hypothesis. One of my students has developed a novel plasma treatment method using pentane, a hydrocarbon precursor. And in her initial experiment, she's varied the time the silk materials are exposed to the plasma. So from anywhere from 1 to 5 minutes of exposing the plasma or exposing the silk film to the pentane plasma. What she's found is that increasing the treatment time yields a more hydrophobic surface. So we're looking again at contact angle results for the untreated film, a short treatment time and a longer treatment time. And our hypothesis is that the coating hydrophobicity is dictated by, partially at least, in part by the conformality of the coating or the uniformity of the coating on the surface that results from the pentane plasma interacting with the surface. Another interesting result that she's seen is that if the films are treated for too long, the contact angle that are measured are actually lower than that of the untreated film, which is very interesting and suggests that there may be competitive etching when the film is exposed to the plasma for increased duration. And we're currently characterizing the films using scanning electron microscopy to see if we can observe any changes in morphology that may arise from this prolonged exposure. And of course, we're also very interested in creating these in vitro enzymatic degradation environments so that we can age the different plasma modified samples and observe the resulting any resulting differences in terms of their enzymatic degradation. So hopefully during today's story, I've convinced you a bit of the importance of biomedical devices and for polymers, for the use of polymers in those devices, and moreover, really the need to modify those devices. So using a material that has it's very challenging to find a material that has both optimal bulk properties and optimal surface properties at the same, in the same material. So oftentimes we need to take a material with favorable bulk properties and modify its surface to induce a particular type of outcome in when that material interacts with the biological environment. So I definitely need to acknowledge two of my students whose results I showed today. These are my students, Ethan and Alyssa. This is, of course, a pre-COVID photo. These days we're 6 feet apart wearing masks in the lab. I also need to acknowledge funding sources from CSUSM and from my department and college for all of their support. I encourage you to follow our progress as we continue to build our lab on our website by scanning this QR code. We're also following me on Twitter. And if you'd like to contact me for more information, feel free to email me at any time. So thank you for your participation and attention at this late hour and right after daylight savings time. And I'll be happy to take any other questions.
Thank you. That was so interesting.
Thanks.
Does anybody have any questions?
So let me jump in too. If you have a question on YouTube, I actually have 3 already, but I will let people in here. If you have a question, please email fall.webinar.questions@gmail.com. It should be in the information under the YouTube link.
Looks like Tamara has a question. Go ahead.
Hi again. Okay, my question is in regards, I guess, to your overall purpose and goals for your specific project. What, in your opinion, are the most important biological factors to consider when you are trying to design your system?
Trying to combine our polymer. So do you mean in terms of like what specific interactions we're trying to control?
I guess like one other similar type polymer development that I had recently looked up was in regards to adhesives, polymers adhesives that wet set within the body. And 2 of the things that they had listed in their studies that were important to consider were acidity and salinity. And so things like that would affect negatively or inhibit your polymer in any way. What are you guys thinking or looking at?
Got it. No, that's a good consideration for sure. And luckily or not luckily, however you think about it, we're generally using polymers that are already pre-formed in terms of we're already relying on these solid constructs or creating these solid constructs with particular geometries or architectures. to then modify for kind of the next use. So we don't need to worry too much, at least for our initial interactions. We're generally having, that solid aqueous interface. So a lot of those parameters, like what you had mentioned, they do play a role, but not in so much as we're dealing with an aqueous phase polymer in the body. So that's definitely a more complex system.
Okay.
What I would say though, is that when we're creating materials and we're trying to test them to change or to evaluate, interactions in an in vitro setting, since that's mainly what we plan to be using, we're definitely controlling factors like humidity, salinity, and temperature in terms of how we're designing those in vitro environments to not, it's impossible to 100% recapitulate the biological environment, but I would say that those factors are definitely critical to control in terms of looking at how a particular biological species will interact with the solid surface.
Awesome, thank you.
You're welcome.
Our next question is from Dr. Schmidt.
Yeah, with your graduate work with the caprolactone constructs there, I was impressed with the high contact angle. And I was wondering how much of that is due to the nanostructure of the surface versus the actual hydrophobicity of the material.
Yeah, that's an excellent point. Basically, contact angles of greater than 90 degrees or so typically are arising from some kind of inherent morphology, whether it be nano or microstructure of the surface. So flat polycaprolactone films that we created with the same molecular weight polymer have contact angles around 75 degrees, so still considered to be hydrophobic, but the major or a large part of that comes from that porous structure. Absolutely.
And we have a question from YouTube.
Yes, Morgan. So fantastic talk. And I think I want to put my little spin on this too. I can't remember the last time I've played with plasma. How safe is this procedure? Because I've never done anything like this before.
Yeah, so we use an alternating current power supply, typically at relatively low applied powers, so between 25 and 50 watts. So aside from physically touching the induction coil, there's very little that one could do to make the process dangerous. Of course, you could imagine, we are using pressurized gases and glassware. So anytime those things are combined, there are inherent risks. We actually operate our plasma reactor in a fume hood. So we have the fume hood sash closed during operation, which definitely reduces a lot of risk associated with vacuum and glass combinations. And the other kind of point that I'd want to mention with safety depends on the precursor. So if, for example, if we were using something like nitric oxide, it's very critical to have a proper ventilation system with the pump and everything like that. For other types of, or for certain precursors, we also use a liquid nitrogen trap to keep that out of the pump. So we have some or several, I would say, mechanisms to enhance the safety, but of course there's some inherent risk.
That's terrific. And I have a follow-up that's related. What scale would this be done in industry? How does this relate to large scale?
Yeah, it actually is done in a lot of different industries. So most common of which is the semiconductor industry. So creating silicon chips uses plasma in many different ways. It can be, depending on the plasma conditions, it can be used as like a translating plasma application device where it will go over multiple, like a large batch of substrates, which are in those cases generally silicone wafers, to result in a uniform treatment. So there are definitely industrial scale processing devices that do utilize plasma. And it may be in a larger reactor, either with a translating electrode or in a large scale dielectric barrier discharge setup, which is a little different than the one that we use in our lab, but effectively the substrate sits on one of the electrodes, and that could be done on a fairly large scale. Definitely like in the meter or larger length in terms of reactor.
Awesome, thank you.
Yeah.
I was wondering what some of the common gases that you use in your lab are for precursors. I know you mentioned that you use essential oils for.
Yeah, so we don't currently use essential oils, but they have been used in similar plasmas. One of the people in my graduate group used the eucalyptus essential oil, cineol, as a plasma precursor that effectively deposited this kind of hydrocarbon containing functional film. We currently are using pentane, ethylene, water vapor, and we also use oxygen. And the reason for that is because oxygen, we don't generally use it to treat surfaces, although it is very commonly used to treat surfaces. It's one of the most common plasma precursors. We actually use it for cleaning. So if you imagine we're flowing this pentane precursor in, we're depositing a hydrocarbon film on everything, not just the surface of the film, the silk film, but also the walls of the reactor. And if we then were to use a different precursor like water vapor, we would have some contaminants that would be coming effectively from getting etched off of the walls, which could then cause inconsistencies in our surface. So we use oxygen plasma as a cleaning method in between different plasma treatments, which effectively etches all of that hydrocarbon precursor off of the walls and creates effectively a clean surface for us to then use a different precursor. So it works pretty well.
That's really interesting. Just like the thought of using that to attach different things. It's really cool.
Thank you.
Are there any more questions? I also was wondering, oh, sorry, go ahead.
There's a couple more from YouTube. You've really inspired some people, Morgan. So another question, have you seen temperature influence the hydrophobicity? I think they mean it showed up when you were showing the time delay for degradation. So does temperature affect I think before, one more slide.
This one?
Yeah, that's when it showed up. Mm-hmm.
Oh, okay. We haven't looked at aging the films in different temperature conditions. That's definitely something we're interested in. We're also interested in aging them. So temperature is one variable. We're also interested in aging them in aqueous conditions. So if we take these silk films and age them in an aqueous environment, and then take them out and dry them, are their surface properties changing after exposure to that aqueous environment? So we're definitely looking into that, but we haven't measured it yet.
And then one more question from YouTube. You look like you're doing polymer and bio and inorganic. What do you consider yourself?
That's a great question. So I like to think that I don't really fit in any of those boxes. I think chemistry and science in general, but chemistry especially, is really evolving into a field where multidisciplinary or transdisciplinary work is really the path forward. So I use a lot of different techniques in my research that are inspired by materials chemistry and biochemistry, biology. and biomedical engineering. So those may be some boxes. Luckily, as a material scientist, we don't really have to check any boxes because material science draws on so many other disciplines. But I think that's really a positive feature of the field of material science, is that it draws on so many other disciplines and really works to combine knowledge from these different fields to kind of move the entire biomedical space forward. So it's really exciting. It's a really great field to be in. I think I answered that question by not answering it, but hopefully everybody is.
I'll send an apology email. It's good to not check the boxes, though. I think that's pretty cool.
For sure.
Daniel has a question.
Yeah, thank you for your talk. I had a question about the instrument you said you use, that you put a drop of water on it and then it gives you the angle. Like how big is it? Is it kind of like takes up a bunch of room like an HPLC? Like how long is the runtime and what other applications does it have? Oh, and is that the image that it gives out, like the little video you have?
Yeah, so the instrument is called a contact angle goniometer. It probably has a footprint of around 2 feet by 1 foot, about. Pretty compact. The major features is that it has a stage, a camera, a backlighted feature, and then it's connected to a software package that actually does the physical fitting of the drop profiles. So we can obtain still images, we can obtain dynamic videos, or more complicated types of surface analyses where we're looking at advancing and receding. So imagine that they take the water drop, and instead of just dropping it onto the surface, we actually increase the volume and decrease the volume, and we can measure those different angles, which actually tell us something about the surface. So the major application of contact angle goniometry is to measure the surface wettability, which is connected to the free energy of the surface. So another possibility is to use multiple probe liquids. So we could use polar and non-polar probe liquids to see the differences in how those liquids interact with the surface, and do some modeling to approximate the surface free energy of the material prior and after plasma modification. We're right now sticking to water because we're interested in looking at these things in a biological environment, obviously aqueous. But yes, it looks like somebody in the chat said they made a crude one. Yes, it can be done. It's possible to make any kind of measurement like this if you have a syringe that you can create a dosing of with a particular drop size, so that you can tune that, then add the drop to the surface. And if you have some type of camera, it doesn't need to be fancy, or microscope, it's possible to then capture that image, and then the drop profile can be measured or approximated using kind of some by hand modification or measurements. So it can be very technologically advanced or very simple, but the best thing about it is that it's the most surface sensitive technique there is. We're talking about a liquid interacting with the top monolayer of a surface, which is more surface sensitive than almost any other type of characterization technique. So very powerful.
Okay. Thank you. And Dr. Schmidt, I'm gonna have to stop by your lab to check that out. Thanks.
And our last question will be from Karen.
Hi, thank you so much for that talk. The moment you started off with asking the question about the contacts, even though I don't wear contacts, it still was very intriguing. So just that's an amazing hook. You will definitely get people listening right away because it's relatable.
Thank you.
So, kind of piggybacking on Tamara's question about kind of carrying out like testing on these, I guess you could say, applications for biomedical devices. So you mentioned about the in vitro environment and kind of doing that, like first feeling it out with that. So obviously, in vivo, sometimes could be a different environment. Is there some type of studies that you found or something that you are particularly are afraid of when it comes to now testing it and applying it to, human interaction?
Sure. Yeah, that's always the kind of pie in the sky type of question. So I like to think that the work that we do is really more of even though some people in the chemistry field would call it applied research, I really think about it as really basic research, and that we're really looking at a particular type of surface modification and how that influences the surface properties of the polymer. When it comes down to it, the majority of interactions that happen the second a foreign material is introduced into the body is that material gets coated in protein. That's kind of the first step. So whether that's favorable or not will depend on the application, but it's almost an inherent truth, depending on the surface modification. So that's definitely something that I like to think about and keep in the back of my head, even though we want to promote an interaction, let's say, between a surface and a fibroblast or a particular type of, bacteria, or we want to reduce the adhesion of blood cells. Like, all of those are actually governed by this protein adsorption, this protein layer, and it's a very complex system. So keeping in mind the type of interaction we're shooting for, but also while appreciating the complexity of the biological environment is something that's always at the forefront of my mind. I'm not sure I'd necessarily say I'm worried about it, because it's just kind of an inherent, part of the field, but it's definitely something that I think about and try to remind myself of.
Oh, can I ask one little follow-up question about that?
Sure.
You also mentioned, of course, that there's some people that have, extreme like dryness in their eyes and so forth. Is that something that you think would be worked towards like a universal kind of suit, or would it be more for a specific population and then a different product for other people?
Yeah, I think that there are a lot of possibilities with respect to the modifications that can be made to the material, and a lot of room for customization for a specific type of biological environment. I think there is definitely a lot of interest in the field to create those types of customizable materials. And I definitely like to think that plasma treatment is one method for which we can do that, because it's a very high throughput technique where making small changes to the parameters can have large changes to the surface properties. So it would be at least relatively straightforward to evaluate and to prepare surfaces with these multitudes of characteristics, for sure.
Well, thank you so much for that.
Yeah, thank you.
So can everyone please join me in thanking Dr. Hawker. Thank you so much I definitely learned a lot about polymers, plasma reactors, biomedical devices. I now see all the importance in them. So thank you very much. It was really, really insightful.
Of course. Thank you so much for having me. This has been great.